Tissue culture. It refers to the removal of cells from an animal or plant and their subsequent growth in a favorable artificial environment. The cells may be removed from the tissue directly and disaggregated by enzymatic or mechanical means before cultivation, or derived from a cell line or cell strain that has already been established. Types of cell culture. Organ culture. Small bits of organs can be maintained in vitro for days and weeks, preserving their original architecture and function, useful in isolation of some viruses. Explant culture. Fragments of minced tissue can be grown as explants embedded in plasma clots, may also be cultivated in suspension. This method is now seldom employed in virology. Cell culture. This is the type of culture routinely employed for growing viruses. Tissues are dissociated into component cells by the action of proteolytic enzymes, such as trypsin and mechanical shaking. The cells are washed, counted, and suspended in a growth. The essential constituents of the growth are added. The cell suspension is dispensed in bottles, tubes, or petri dishes. The cells adhere to the glass surface and on incubation, divide to form a confluent monolayer sheet of cells covering the surface within about a week. Cell culture tubes are incubated. Cell culture classified into three types. 1. Primary cell culture. These are normal cells freshly taken from the body and cultured. They are capable of only limited growth in culture and cannot be maintained in serial culture. Primary cell cultures are useful for the isolation of viruses and their cultivation for vaccine production. Diploid cell strains. These are cells of a single type that retain the original diploid chromosome number and karyotype during serial subcultivation for a limited number. Continuous cell lines. These are cells of a single type, usually derived from cancer cells that are capable of continuous serial cultivation indefinitely.